Good evening to each and every one of you present here today. We will be delving into the topic of respiratory physiology tonight. So a summary of today's presentation is reflected here. So firstly, just a quick anatomy recap. The respiratory system is composed primarily of the nose, oropharynx, larynx, trachea, bronchi, bronchioles, and lungs. The lungs are the foundational organs of this system, whose most basic function is to facilitate gas exchange from the environment into the bloodstream. They are paired, spongy, coney-shaped organs located in the thoracic cavity, occupying the right and left pleural cavities separated by the mediastinum. The varying parts are listed here along with the vascular supply. The right lung, shorter, wider, and more vertical, consists of 3 lobes, 2 fissures, and 10 bronchopulmonary segments, whilst the left lung consists of 2 lobes, 1 fissure, a cardiac notch, and a lingular, and 8 to 10 bronchopulmonary segments. Each bronchopulmonary segment is a functionally independent unit of the lung supplied by one segmental tertiary bronchus, one segmental pulmonary artery branch, and drained by intersegmental pulmonary veins. The region of the mediastinum where the lung is connected to the trachea and heart is the root of the lung, and the region of where the bronchi and pulmonary vessels enter or leave is referred to as the hilum. The pleura, a thin fibrous tissue membrane surfaced by mesothelial cells, clothes each lung and lines the thoracic cavity. It's composed of the viscera pleura lining the lung and the parietal pleura lining the thoracic wall, diaphragm, and mediastinum. The pleural cavity contains a thin film of pleural fluid which reduces friction, maintains negative intrapleural pressure, and allows smooth lung expansion. On the right illustrates the conducting and respiratory zones. And the alveoli are the functional units for gas exchange, and they are type 1 and 2 pneumocytes. The former cover 95% alveolar surface and are thin squamous cells, whilst the latter produce pulmonary surfactants and can proliferate and replace damaged type 1 cells. The alveolar capillary membrane consists of alveolar epithelium, fused basement membrane, and capillary endothelium. Oxygen gets transported through the alveoli into the capillary network, where it can enter the arterial system, ultimately perfusing tissue. So the main functions of respiration are oxygen provision to the tissues and carbon dioxide removal. The 4 major components are as follows: pulmonary ventilation, diffusion of oxygen and carbon dioxide between the alveoli and blood, transport of oxygen and carbon dioxide in the blood and body fluids to and from the body's tissue cells, and regulation of ventilation. Figure 38, on the right, shows 2 methods of lung expansion and contraction. 1, by downward and upward movements of the diaphragm to lengthen and shorten the chest cavity, and 2, by elevation or depression of the ribs to increase or decrease the AP diameter of the chest cavity. Normal quiet breathing is accomplished almost entirely by movement of the diaphragm, and during inspiration, contraction of the diaphragm pulls the lower lung surfaces downward in an active process. The contraction of the inspiratory muscles increases intrathoracic volume. In expiration, the diaphragm simply relaxes, and the elastic recoil of the lungs, chest wall, and abdominal structures compresses the lungs, expelling the air. The second method for lung expansion is raising the rib cage. When the rib cage is elevated, the ribs project almost directly forward, allowing the sternum to also move forward, away from the vertebral column, increasing the AP chest thickness by about 20% greater during maximum inspiration than during expiration. The various muscles elevating the chest cage, that is the muscles of inspiration, and the muscles that depress it, the muscles of expiration, are shown here. Figure 38.1 also shows the mechanism whereby the external and internal intercostals act to cause inspiration and expiration. During expiration, with the ribs angled downward, the external intercostals are elongated forward and downward. As they contract, they pull the upper ribs forward in relation to the lower ribs, causing leverage on the ribs to elevate them, causing inspiration. The internal intercostals angle between the ribs in the opposite direction and cause the opposites. The lung floats in the thoracic cavity surrounded by pleural fluid. Continual suction of excess fluid into lymphatic channels maintains a slight suction between the visceral and parietal pleural surfaces. Therefore, the lungs are held to the thoracic wall as if glued, but are well lubricated and can slide freely as the chest expands and contracts. Pleural pressure is the pressure of the fluid in the thin space between the lung and chest wall pleura, and it's normally a slightly negative pressure. At the beginning of inspiration, normal pleural pressure is negative 2.5 to 5 centimeters of water, the amount of suction required to hold the lungs open to their resting level. During normal inspiration, expansion of the chest cage pulls outward on the lungs with a greater force and creates a more negative pressure of around negative 6 to negative 7.5 centimeters of water. At the end of inspiration, the lung recoil pulls the chest back to the expiratory position, where the recoil pressures of the lung and chest wall balance. These relationships between pleural pressure and changing lung volumes are demonstrated in figure 38.2. The lower panel shows the increasing negativity of the pleural pressure during inspiration, and in the upper panel, an increase in lung volume of about 0.5 liters. During expiration, the events are reversed. Expiration during quiet breathing is passive, no muscles contract, decreasing in thoracic volume. However, some contraction of the inspiratory muscles occurs in the early parts of expiration, which exerts a braking action on the recoil forces and slows expiration. When the glottis is open and there is no airflow, pressures in all parts of the respiratory tree, including to the alveoli, are almost equal to atmospheric pressure, which is zero reference pressure, that is 0 centimeters of water. For inward airflow into the alveoli during inspiration, the pressure in the alveoli must fall slightly below atmospheric pressure. The second curve on figure 38.2, labeled alveolar pressure, demonstrates during normal inspiration, alveolar pressure decreases to negative 1 centimeter of water, which is enough to pull 0.5 liters of air into the lungs in 2 seconds required for normal quiet inspiration. During expiration, alveolar pressures rise to plus 1 centimeter of water, forcing this 0.5 liter of inspired air out of the lung during the same time. Also in figure 38.2, the transpulmonary pressure is the pressure difference between that in the alveoli and on the outer surfaces of the lung, or pleural pressure, and is a measure of the elastic forces in the lung that tend to collapse the lungs at each instance of respiration, called the recoil pressure. The extent to which the lungs will expand for each unit increase in transpulmonary pressure is called the lung compliance. The total compliance of both lungs together in the normal average adult averages around 200 mils of air per centimeter of water transpulmonary pressure, meaning every time the transpulmonary pressure increases by 1 centimeter of water, the lung volume after 10 to 20 seconds will expand by 200 mils. Figure 38.3 is a compliance diagram of the lungs relating lung volume changes to changes in pleural pressure, which in turn alters transpulmonary pressure. Note that the relationship is different for inspiration and expiration, with the two curves being called inspiratory compliance curve and expiratory compliance curve. Each curve is recorded by changing the pleural pressure in small steps, allowing the lung volume to come to a steady level between successive steps. The characteristics of the compliance diagram are determined by elastic forces of the lung, which can be divided into two parts. The elastic forces of the lung tissue, determined mainly by elastin and collagen fibers interwoven among lung parenchyma, and elastic forces caused by surface tension of fluid lining the alveoli inner walls and other lung spaces. 38.4 shows the significance of surface tension using a lung compliance diagram, comparing when filled with saline solution and then with air. When air-filled, there is an interface between the alveolar fluid and air in the alveoli. In saline-filled lungs, there is no air-fluid interface and therefore no surface tension effect. Only tissue elastic forces are at operation. Also, transpleural pressures required to expand air-filled lungs are around 3 times as great as those required to expand saline-filled lungs. Thus, tissue elastic forces tending to cause collapse of the air-filled lung represent only 1/3 of the total lung elasticity, versus the air fluid surface tension forces in the alveoli represent 2/3. So airway resistance is defined as the change of pressure from the alveoli to the mouth divided by the change in flow rate. Measurements comparing alveolar, intrapleural, and actual pressure shown in figure 34.7 in the middle panel, which shows the contribution of airway resistance. Airway resistance is significantly increased as lung volume is reduced. Also, the bronchi, bronchioles, and contraction of smooth muscles that line the bronchial airways all increase airway resistance. An important factor affecting lung compliance is alveolar surface tension. The magnitude of this component at various lung volumes can be measured by distending the lungs alternately with saline and with air while measuring the intrapulmonary pressure. Because saline reduces the surface tension to nearly zero, the pressure volume curve obtained with saline measures only the tissue elasticity, as in figure 3412, versus when the curve with air measures both tissue elasticity and surface tension. The difference between the curves is much smaller when the lung volumes are small. Differences are also obvious in the curves generated during inflation and deflation, and this term is called hysteresis and is not present in the saline generated curves. The alveolar environment and secreted factors that help reduce surface tension and keep the alveoli from collapsing contribute to hysteresis. So we know surfactant as most important. At birth, the fetus makes respiratory movements in utero, but the lungs remain collapsed until birth. After birth, the infant makes several strong inspiratory movements and the lungs expand. Surfactant keeps them from collapsing again. It's a mixture of dipalmitoyl phosphatidylcholine, or DPPC, other lipids and proteins, and it's due to the presence of same that the surface tension is low when the alveoli are small. If the surface tension is not kept low when the alveoli become smaller during expiration, they collapse in accordance with the law of Laplace, which states in spherical structures such as the alveolus, the distending pressure equals 2 times the tension divided by the radius. So if the tension is not reduced as the radius is reduced, the tension overcomes the distending pressure. Surfactant also helps prevent pulmonary edema, and if it weren't present, the unopposed surface tension in the alveoli would produce about a 20 millimeters of mercury force, greatly favoring transudation of fluid from the blood into the alveoli. Formation of the phospholipid film is greatly facilitated by proteins in the surfactant, and this material contains 4 unique ones, the surfactant proteins A, B, C, and D. So modern spirometers permit direct measurements of gas intake and output. After expiration, during quiet breathing, the lungs have a tendency to collapse and the chest wall has a tendency to expand. The interaction between the lung recoil and chest recoil can be measured through a spirometer that has a valve just beyond the mouthpiece, on which after the person inhales a given amount, the valve is shut, closing off the airway, the mouthpiece containing the pressure measuring device, which records the airway pressure while the respiratory muscles relax. The procedure is repeated after inhaling or actively exhaling various volumes. The curve of airway pressure obtained in this way, plotted against volume, is the pressure volume curve of the total respiratory system, or PTR, shown in 3410. The pressure is 0 at a lung volume that corresponds to the volume of gas in the lungs at the functional residual capacity, or relaxation volume. Figure 3410, this relaxation pressure is the sum of a slightly negative pressure component from the chest wall, or PW, and a slightly positive pressure from the lungs, or PL. The PTR is positive at greater volumes and negative at smaller volumes. Lung and chest wall compliance is measured as a slope of the PTR curve, or as a change in the lung volume per unit change in airway pressure. Compliance is also slightly greater when measured during deflation than during inflation. The curve is shifted downward and to the right by pulmonary edema and interstitial pulmonary fibrosis as compliance is decreased, versus the curve is shifted upward and to the left in emphysema where compliance is increased. So important quantification of lung function can be gleaned from the displacements of air volume during inspiration and or expiration. Lung capacities refer to subdivisions that contain 2 or more volumes. Volumes and capacities recorded on a spirometer are shown in figure 34.8. The amount of air that moves into the lungs with each inspiration or out with each expiration during quiet breathing is called the tidal volume, or TV. Typical values around 500 to 750 milliliters. The air inspired with maximal inspiratory effort in excess of the TB is the inspiratory reserve volume, which is typically around 2 liters. The volume expelled by an active expiratory effort after passive expiration is the expiratory reserve volume, typically around 1 liter ERV. The air left in the lungs after maximal expiratory effort is a residual volume around 1.3. When all of the above 4 components are taken together, they form the total lung capacity, accounting for about 5 liters. then the total lung capacity can be broken down into alternative capacities that help define functioning lungs. The vital lung capacity, or VLC, refers to the maximum amount of air expired from the fully inflated lung, or the maximum inspiratory level, represented by TV plus IRV plus ERV. The inspiratory capacity, or IC, around 2.5 liters, is the maximum amount of air inspired from the end expiratory level, or IRV plus TV, and the functional residual capacity represents the volume of air remaining in the lungs after expiration of a normal breath. Dynamic measurements of lung volumes and capacities aid in determining the lung function. The forced vital capacity, the largest amount of air that can be expired after maximal inspiratory effort, is frequently measured clinically as an index of pulmonary function, informing about the strength of the respiratory muscles and other aspects. As shown in figure 34.9, the fraction of vital capacity expired during the first second of a forced expiration is referred to as FEV1. The FEV1 to FVC ratio is a useful tool in recognizing classes of airway disease. In the example shown to the right on the bottom, a healthy FVC is around 4 liters, and a healthy FEV1 is around 3.3 liters, with a calculated ratio is about 80%, 0.8. Patients with obstructive or restrictive diseases can display a reduced FVC of about 2 liters, but FEV1 measurements vary between two diseases. In obstructive disorders, patients tend to show a slow, steady slope to the FVC, resulting in a small FEV1, such as 1 liter in the example. However, in the restrictive disorder, patients' airflow tends to be fast at first and then quickly levels out to approach the FVC. The resultant FEV1 is much greater, around 1.8, even though the FVC is equivalent. A calculation of the FEV1 and FVC for patients with obstructive is about 50% versus restrictive as 90%, and this defines the hallmark measurements in evaluating these two diseases. Obstructive disorders result in a marked decrease in both FVC and FEV1/FVC ratio, whereas restrictive disorders result in a loss of FVC without the loss in FEV1 to FVC. However, it should be noted that these examples are idealized and several disorders could show mixed readings. Other dynamic measurements include the respiratory minute volume, or RMV, and the maximal voluntary ventilation, MVV, with the former normally about 6 liters, and the latter being the largest volume of gas that can be moved into or out of the lungs in 1 minute by voluntary effort, typically being measured over 15 seconds and prorated to a minute. Normal values are about 140 to 180 liters per minute, and changes in the RMV and MVV in a patient can be indicative of lung dysfunction. So work is performed by the respiratory muscles in stretching the elastic tissues of the chest wall and lungs by elastic work, accounting for about 65%, moving inelastic tissues against viscous resistance, about 7%, and moving air through the respiratory passages against airway resistance, about 28%. Because the dimensions of pressure by volume has the same dimensions as work, force by distance, grams by centimeters, the work of breathing could be calculated from the previously presented pressure volume curve in figure 3410. Estimates of the total work of quiet breathing range from about 0.3 to 0.8 kilograms per meter per minute, and these values rise markedly during exercise, but the energy cost of breathing is around less than 3% of the total energy expenditure. The work of breathing is greatly increased in diseases such as emphysema, asthma, heart failure with dyspnea and orthopnea, and the respiratory muscles have a length tension relations like those of other skeletal and cardiac muscles, and when they are severely stretched, they contract with less strength, can also become fatigued and fail, leading to inadequate ventilation. Now, in the upright position, ventilation per unit lung volume is greater at the base than apex because at the start of inspiration, the intrapleural pressure is less negative at the base than apex. And since the intrapulmonary intrapleural pressure difference is less than at the apex, the lung is less expanded. Conversely, at the apex, the lung is more expanded and the percentage of maximum lung volume is greater. Because of the stiffness of the lung, the increase in lung volume per unit increase in pressure is smaller when the lung is initially more expanded and the ventilation is consequently greater at the base. Blood flow is also greater at the base. The relative change in the blood flow from the apex to the base is more than the relative change in the ventilation. So the ventilation perfusion ratio is low at the base and high at the apex. Now, traditionally, these differences were thought to be caused by gravity. Gravity pulls more blood to the base of the lungs. The base of the lungs also creates a pressure difference affecting ventilation. However, new evidence regarding studies of astronauts in weightlessness showed that differences in ventilation and blood flow still exist even without gravity. Therefore, gravity is not the only cause. Other factors such as lung structure and branching patterns of the airways and blood vessels also contribute to these regional differences. Because gaseous exchange occurs only in the terminal airway portions, the gas that occupies the remaining respiratory system isn't available for exchange with pulmonary capillary blood. Normally, the volume in moles of this anatomic dead space is about equal to the body weight in pounds. For example, in a 150-pound or 68 kg man, only the first 350 mils of 500 mils inspired with each breath at rest mixes with the air in the alveoli. Conversely, with each expiration, the first 150 mils expired is gas that occupied the dead space, and only the last 350 mils is gas from the alveoli. Consequently, the alveoli ventilation, or the amount of air reaching the alveolar per minute, is less than the respiratory minute volume, or less than the 500 mils of breath by 12 breaths per minute, which is equal to 6 liters. Because of the dead space, rapid, shallow breathing produces much less alveolar ventilation than slow, deep breathing at the same RMV, shown in table 34.1 here. It's important to distinguish between anatomic dead space, which is the respiratory system volume exclusive of the alveoli, versus total physiologic dead space, which is the volume of gas not equilibrating with blood, and that is the wasted ventilation. So in healthy individuals, the two dead spaces are identical and can be estimated by body weight. In disease states, no exchange may take place between the alveoli gas and blood, and some of the alveoli may be overventilated. The volume of gas in a non-perfused alveoli and any excess volume of alveoli air constitutes dead space non-equilibrating gas volume. The anatomic dead space could be measured by analysis of a single breath nitrogen curves seen in figure 34.13. From mid-inspiration, the patient takes a deep breath of pure oxygen, exhaling steadily while the nitrogen content of the expired gas is continuously measured. The initial gas exhaled, or phase 1, is the gas that fills the dead space and that consequently contains no nitrogen. This is followed by a mixture of dead space and alveolar gas, or phase 2, and then by alveolar gas, or phase 3. The volume of dead space is the volume of the gas expired from peak inspiration to the midpoint of phase 2. Phase 3 of the single breath nitrogen curve terminates at the closing volume, which is the lung volume above the residual volume, at which the airways in the lower dependent parts of the lung begin to close off because of lesser transmural pressure. The gas in the upper lung is richer in nitrogen than the gas in the lower dependent portions of the lung because the upper alveoli are more distended at inspiration start and the nitrogen is less diluted with oxygen. Phase 3 has a slight positive slope even before phase 4, indicating a gradual increase in the expired gas proportion of the expired gas from the nitrogen-rich upper lungs. Phase 4 follows, during which the nitrogen content of the expired gas is increased. The total dead space could be calculated using Bohr equation, shown here, which states the tidal volume times the partial pressure of expired carbon dioxide equals the partial pressure of arterial carbon dioxide times the difference between the tidal volume and dead space, plus the partial pressure of inspired carbon dioxide times the dead space. However, since the PiCO2 times the BD is so negligible, the equation for dead space could be solved as shown below. Now, the pressure exerted by any one gas in a mixture, that is its partial pressure, is equal to the total pressure times the fraction of the total amount of the gas it represents. So the pressure, or P, of a gas is proportional to its temperature, or T, time, and the number of moles, N, occupying a certain volume, V, as seen in table 34.2. Dry air's composition is about 20.9% oxygen, 0.04% carbon dioxide, 78% nitrogen, and 0.9% other inert constituents. The barometric pressure at sea level is around 760 millimeters of mercury, or 1 atmosphere. The partial pressure of oxygen in dry air is therefore around 160 millimeters of mercury at sea level, and the partial pressure of nitrogen and other inert gases around 600 millimeters of mercury, and that of carbon dioxide around 0.3. Water vapor in the air reduces these percentages and therefore the partial pressures. So air equilibrated with water is saturated with water vapor and along with the inspired air before it reaches the lungs. The partial pressure of water at body temperature around 37 degrees Celsius is around 47 millimeters of water. And we know that gas diffuses from areas of high pressure to areas of low pressure with diffusion rate depending on the concentration gradient and the nature of the barrier between the two areas. So figure 34.12 compares the composition of 34.14, compares the composition of alveolar gas with inspired and expired air. The partial pressure of oxygen in arterial blood could also be calculated from the alveolar gas equation shown here, where the PiO2 is the partial pressure of inspired O2. The partial PaCO2 is the partial pressure of inspired CO2. The FiO2 is the fraction of oxygen molecules in dry gas, and the R is the respiratory exchange ratio. That is the flow of carbon dioxide molecules across the alveolar membrane per minute. So in the steady state, inspired air mixes with alveolar gas, replacing the oxygen that has entered the blood and diluting the carbon dioxide that has entered the alveoli, with parts of this mixture being expired. The oxygen content of the alveolar gas then falls and the carbon dioxide content rises until the next inspiration. So blood takes approximately 0.75 seconds to traverse the pulmonary capillaries at rest, and substance uptake can be limited depending on the interactions between substances in blood, preventing the passage from the alveoli to the capillary blood reaching equilibrium. Namely, anesthetic gas nitrous oxide does not react and reaches equilibrium in about 0.1 seconds, as seen in figure 34.15. In this situation, the amount of nitrous oxide taken up is not limited by diffusion, but by the amount of blood flowing through the capillaries. Therefore, it is flow limited. On the other hand, carbon monoxide is taken up by hemoglobin in the red blood cells at such a high rate that the partial pressure of carbon monoxide in capillaries stays very low, and the equilibrium is not reached in 0.75 seconds. Therefore, the transfer is diffusion limited. And the oxygen is taken up by hemoglobin, but much less avidly than carbon monoxide, and reaches equilibrium in around 0.3 seconds. Thus, its uptake is perfusion limited. So the diffusing capacity of the lung for a given gas is directly proportional to the alveolar capillary membrane surface area and inversely proportional to its thickness. The diffusing capacity of carbon monoxide, or DLCO, is measured as an index of diffusing capacity because its uptake is diffusion limited, and it's proportional to the amount of carbon monoxide entering the blood divided by the partial pressure of carbon monoxide in the alveoli, as shown here. This at rest is around 25 mils per minute per millimeter of mercury, increasing up to threefold during exercise due to capillary dilation and increasing numbers of active capillaries. The partial pressure of alveolar oxygen is normally about 100 millimeters of mercury, and the partial pressure of pulmonary capillary oxygen is around 40 millimeters of mercury. The partial pressure of arterial oxygen is around 97, just under that of alveolar oxygen. So the diffusing capacity for oxygen, like that of carbon monoxide at rest, is around the same, 25. And the partial pressure of carbon dioxide of venous blood is around 46, whereas that of alveolar air is around 40 millimeters of mercury. And carbon dioxide diffuses from the blood into the alveoli along this gradient. So carbon dioxide passes through all biological membranes with ease, and its diffusing capacity of the lung is much greater than that of oxygen. It's for this reason that carbon dioxide retention is really a problem in alveolar fibrosis, even when the reduction in diffusing capacity of oxygen is severe. Right, so regarding the pulmonary circulation, the pulmonary artery walls and its branches are about 30% as thick as the wall of the aorta, and the small arterial vessels are endothelial tubes with a little muscle in their walls. The postcapillary walls contain some smooth muscle, and the pulmonary capillaries are large with multiple anastomoses, and each alveolus sits in a capillary basket. With two minor exceptions, the blood expelled by the left ventricle returns to the right atrium and is ejected by the right ventricle, with the pulmonary vasculature accommodating a blood flow almost equal to that of all organs in the body. One exception is part of the bronchial blood flow. There are anastomoses between the bronchial capillaries and pulmonary arch capillaries and veins. And although some bronchial blood enters the bronchial veins, some also enters the pulmonary capillaries and veins, bypassing the right ventricle. Another exception is the blood that flows from the coronary arteries into the left heart chambers. Because of the small physiologic shunt created by these two exceptions, the blood in the systemic arteries has a partial pressure of oxygen 2 millimeters of mercury lower than that of the blood equilibrated with the alveolar air, and the hemoglobin saturation is about 0.5% less. The pulmonary system pressure gradient is around 7 millimeters of mercury compared with 90 millimeters of mercury in the systemic circulation. The pulmonary capillary pressure is 10 millimeters of mercury, whereas the oncotic pressure is around 25, so that an inward directed pressure gradient of about 15 millimeters of mercury, that is the difference, keeps the alveolar free of all but a thin film of fluid. When the pulmonary capillary pressure is more than 25 millimeters of mercury, pulmonary congestion and edema result. So the volume of blood in the pulmonary vessels at any time is around 1 liter, of which less than 100 mils is within the capillaries. The mean velocity of blood in the pulmonary artery route is the same as in the aorta, about 40 centimeters per second. And it falls off rapidly and then rises slightly again in the larger pulmonary veins, and it takes a red cell about 0.75 seconds to traverse the pulmonary capillaries at rest and about 0.3 seconds during exercise. So in terms of gravity, in figure 3416, in the upper part of the lung, the blood flow is less, alveolar larger, and the ventilation less than at the base. The pressure in the capillaries at the top of the lungs is close to the alveoli atmospheric pressure. Pulmonary arterial pressure is normally just sufficient to maintain perfusion, but if reduced or if the alveolar pressure is increased, some of the capillaries collapse. Under these circumstances, no gas exchange occurs in the affected alveoli, and they become part of the physiologic dead space. In the mid portions of the lungs, the pulmonary arterial and capillary pressure exceeds alveolar pressure, but the pressure in the pulmonary venules may be lower than the alveolar pressure during normal expiration, so the alveoli are collapsed. Under these circumstances, blood flow is determined by the pulmonary artery alveolar pressure difference rather than the pulmonary artery pulmonary vein difference. Beyond the constriction, blood falls into the pulmonary veins, which are compliant and accommodates the amount of blood that constriction lets flow into them. This has been called the waterfall effect. The compression of vessels produced by alveolar pressure decreases and pulmonary blood flow increases as the arterial pressure increases towards the base of the lung. In the lower portions of the lung, the alveolar pressure is lower than the pressure in all parts of the pulmonary circulation, and blood flow is determined by the arterial venous pressure difference. So the ratio of pulmonary ventilation to pulmonary blood flow for the entire lung at rest is around 0.8. However, marked differences occur in this ventilation perfusion ratio in various parts of the lung, resulting from the effects of gravity, and local changes in this ratio are common in disease. If the ventilation to an alveolus is reduced relative to its perfusion, the partial pressure of oxygen in the alveolus falls because less oxygen is delivered to it, and the partial pressure of the carbon dioxide rises as less carbon dioxide is expired. Conversely, if the pressure is reduced relative to ventilation, the partial pressure of carbon dioxide falls because less carbon dioxide is delivered and the partial pressure of oxygen rises because less O2 enters blood. As noted prior, the ventilation and perfusion in the upright position declines in a linear fashion from the bases to the apices of the lungs. And when widespread, non-uniformity of this ventilation and perfusion in the lungs could cause CO2 retention and lower the systemic partial pressures of arterial oxygen. Now, vein constriction increases pulmonary capillary pressure, and pulmonary artery constriction increases the right heart load. Both active and passive factors affect pulmonary blood flow. Pulmonary vessels have extensive autonomic in the innervation and stimulation of cervical sympathetic ganglia reduces pulmonary blood flow by up to 30%. The vessels also respond to circulating humoral agents, as shown in table 34.3, affecting pulmonary smooth muscle. Many dilator responses are endothelium dependent and presumably operate via the release of nitric oxide. Passive factors, such as cardiac output and gravitational forces, also have significant effects on pulmonary blood flow. With exercise, cardiac output increases with pulmonary arterial pressure rising. More red cells move through the lungs without any oxygen saturation of hemoglobin reduction, giving way to increased total oxygen delivered to the systemic circulation. Capillary dilatation and recruitment of underperfused capillaries occur to carry blood, with the net effect being marked increase in pulmonary blood flow. Now, when a bronchus or bronchiole is obstructed, hypoxia develops in the underventilated alveoli beyond the obstruction, and this oxygen deficiency acts directly on the vascular smooth muscle in the area, producing constriction, shunting blood away from the hypoxic area. Accumulation of the carbon dioxide leads to a pH level drop in the area, producing lung vasoconstriction, as opposed to vasodilation in other tissues. Conversely, blood flow reduction to lung portion lowers the partial pressure of alveolar carbon dioxide, leading to constriction of the bronchi supplying it, shifting the ventilation away from the poorly perfused area. So systemic hypoxia also causes pulmonary arterioles constriction with resultant increase in the pulmonary arterial pressures. In addition to gas exchange, the lungs have a number of metabolic functions, such as the manufacturing of surfactant containing a fibrinolytic system that lyses clots in the pulmonary vessels. They also release a variety of substances, as seen here in 34.4, that enter the systemic arterial blood and remove other substances from the systemic venous blood that arrive via the pulmonary artery. Prostaglandins, for example, are removed from the circulation, but also synthesized in the lungs and are released into the blood when the lung tissue is stretched. The lungs also play an important role in activating angiotensin. The physiologically inactive decapeptide angiotensin 1 is converted to the presaldosterone-stimulating octapeptide angiotensin 2 in the pulmonary circulation, a reaction that occurs in other tissues as well, but is particularly prominent in the lungs. Large amounts of the angiotensin converting enzyme responsible for this activation are located on the surface of the endothelial cells of the pulmonary capillaries. Circulation time through the pulmonary capillaries is less than 1 second, yet about 70% of the angiotensin 1 reaching the lungs is converted to angiotensin 2 in a single trip through the capillaries. Serotonin and norepinephrine removal reduces the vasoactive substances amount from reaching the systemic circulation. However, many others pass through the lungs without being metabolized, such as epinephrine, dopamine, oxytocin, vasopressin. In addition, various amines and polypeptides are secreted by neuroendocrine cells within the lungs. Now, the concentrations of oxygen and carbon dioxide measured as partial pressures change within each lung region as they flow from higher to lower partial pressures. For example, the partial pressure of oxygen is highest in the alveoli upon inspiration and lowest in deoxygenated blood, whereas partial pressure of carbon dioxide is exactly opposite, allowing for oxygen to cross the alveoli and reoxygenate the blood in the pulmonary vasculature, while carbon dioxide leaves the bloodstream and enters the alveoli where it is expired. However, the quantity of these two gases transported to and from the tissues would be grossly inadequate if it weren't for the fact that about 99% of the oxygen that dissolves in the blood combines with the oxygen-carrying protein hemoglobin, and about 94.5% of carbon dioxide that dissolves enters into a series of reversible chemical reactions that convert it to other compounds. Now, the presence of hemoglobin increases the oxygen capacity of the blood 70-fold, and the reactions of carbon dioxide increase the blood carbon dioxide content about 17-fold. The volume of oxygen delivered to the systemic vascular bed per minute is the product of cardiac output and arterial oxygen concentration. The ability to deliver oxygen in the body depends on both the respiratory and cardiovascular systems, and delivery to a tissue depends on the oxygen amount entering the lungs, the adequacy of pulmonary gas exchange, the blood flow to the tissues, and the oxygen carrying capacity of the blood. Blood flow to an individual tissue depends on cardiac output, degree of constriction of the vascular bed in the tissue. Now, the amount of oxygen in the blood is determined by the amount of dissolved oxygen, the amount of hemoglobin in the blood, and the affinity of the hemoglobin for oxygen. Hemoglobin is a protein composed of two subunits, each containing a heme moiety attached to a polypeptide chain. In normal adults, most hemoglobin molecules contain two alpha and two beta chains. Heme, as seen in figure 31.7, is a porphyrin ring complex that includes one atom of ferrous iron. Each of the four iron atoms in the hemoglobin can reversibly bind one oxygen molecule. The quaternary structure of the hemoglobin determines its affinity for oxygen. The iron stays in the ferrous state so that the reaction is oxygenation. Because it contains four deoxyhemoglobin units, the hemoglobin molecule can be represented as Hb4, and it reacts with four molecules of oxygen to form Hb4 OH, as seen here, with a rapid reaction occurring in less than 0.01 seconds. In deoxyhemoglobin, the globin units are tightly bound in a tense T configuration, reducing the affinity of the molecule for oxygen. When oxygen is first bound, the bonds holding the globin units are released, producing a relaxed R configuration, exposing more oxygen binding sites, with a net result being around a 500-fold increase in oxygen affinity. In tissues, these reactions are reversed, resulting in oxygen release. And the transition from one state to another occurs about 108 times in a red blood cell's life. Now, the oxygen hemoglobin dissociation curve relates percentage saturation of oxygen carrying power of the hemoglobin to the partial pressure of oxygen, as shown in figure 35.1. This curve has a characteristic sigmoid shape due to the TR configuration and interconversion. Now, combination of the first heme in the Hb molecule with oxygen increases the affinity of the second heme for oxygen, and oxygenation of the second increases the affinity of the third, and so on. And the affinity of the molecule for the fourth oxygen molecule is many times that for the first. So it should be noted that small changes at low partial pressure of oxygen lead to large changes in the saturation of arterial oxygen. When blood is equilibrated with 100% oxygen, the normal hemoglobin becomes 100% saturated. When fully saturated, each gram of normal hemoglobin contains about 1.34 mils of oxygen, and the hemoglobin concentration in normal blood is around 15 grams per deciliter, on average 14 for females, 16 for men. Therefore, 1 deciliter of blood contains around 20 mils of oxygen bound to hemoglobin when the hemoglobin is 100% saturated. Now, in vivo, the hemoglobin in the blood at the pulmonary capillary end is about 97.5% saturated with oxygen. Because of a slight admixture of the venous blood that bypasses the pulmonary capillaries, that is the physiologic shunt, the hemoglobin in systemic arterial blood is only 97% saturated, and therefore contains a total of about 19.8 mils of oxygen. In venous blood at rest, the hemoglobin is 75% saturated, and the total oxygen content is about 15.2 mils per deciliter. Thus, at rest, the tissues remove about 4.6 mils of oxygen for each deciliter of blood passing through them. 3 important conditions affect the oxygen hemoglobin dissociation curve, that is pH, temperature, and the concentration of 2,3-diphosphoglycerate, or 2,3-DPG. A rise in temperature or a fall in pH shifts the curve to the right, as seen in 35.2. When the curve is shifted in this direction, a higher partial pressure of oxygen is required for hemoglobin to bind a given amount of oxygen. Conversely, a fall in temperature or a rise in pH shifts the curve to the left, and a lower partial pressure of oxygen is required to bind a given amount of oxygen. A convenient index for comparison of such shifts is the P50, or the partial pressure of oxygen at which hemoglobin is half saturated with oxygen. The higher the P50, the lower the affinity of hemoglobin for oxygen. Now, the decrease in oxygen affinity of hemoglobin when the pH of the blood falls is called the Bohr effect. And it's closely related to the fact that deoxyhemoglobin binds hydrogen ions more actively than does oxyhemoglobin. The pH of the blood falls as its carbon dioxide content increases. So when the partial pressure of carbon dioxide rises, the curve shifts to the right and the P50 rises. Now, 2,3-DPG is plentiful in red cells and is formed from the 3-phosphoglyceraldehyde, a product of glycolysis via the Embden-Meyerhof pathway, and it's a highly charged anion that binds to the beta chains of deoxyhemoglobin. And one mole of deoxyhemoglobin binds 1 mole of 2,3-DPG. So an increase in 2,3-DPG concentration shifts the curve to the right, causing more oxygen liberation. Because acidosis inhibits red cell glycolysis, the 2,3-DPG concentration falls when the pH is low. Conversely, thyroid hormones, growth hormones, and androgens all cause increase in concentrations of 2,3-DPG and P50. And exercise has also been reported to produce an increase in 2,3-DPG within 60 minutes and P50. Now, at low partial pressure of oxygen values, the oxygen hemoglobin dissociation curve is steep and large amounts of oxygen are liberated per unit drop in partial pressure of oxygen. A contrast to hemoglobin is myoglobin, an iron-containing pigment found in skeletal muscle. It binds 1 rather than 4 oxygen molecules per mole protein, and the lack of cooperative binding is reflected in the myoglobin dissociation curve, which is a rectangular hyperbola rather than the sigmoid curve, seen in figure 35.3. Additionally, the leftward shift of the myoglobin oxygen binding curve demonstrates a higher affinity for oxygen, promoting favorable transfer of oxygen from hemoglobin in the blood, with the steepness of the curve showing that oxygen is released only at low partial pressure of oxygen values, for example, during exercise. The muscle blood supply is compressed during such contractions, and myoglobin can continue to provide oxygen under reduced blood flow and/or reduced partial pressures of oxygen in the blood. Now, the solubility of carbon dioxide in blood is about 20 times that of oxygen, with more carbon dioxide than oxygen present in simple solution at equal partial pressures. The carbon dioxide that diffuses into red blood cells is rapidly hydrated to carbonic acid because of the presence of carbonic anhydrase. As seen in figure 35.4, the carbonic acid dissociates to hydrogen ion and bicarbonates, and the H plus is buffered primarily by hemoglobin, while the bicarbonate enters the plasma. Because deoxyhemoglobin binds more hydrogen ions than oxyhemoglobin and forms carbamino compounds more readily, binding of oxygen to hemoglobin reduces its affinity for carbon dioxide. The Haldane effect refers to the increased capacity of deoxyhemoglobin to bind and carry carbon dioxide. Consequently, venous blood carries more carbon dioxide than arterial blood, with carbon dioxide uptake facilitated in the tissues and its release in the lungs. About 11% of carbon dioxide added to the blood in the systemic capillaries is carried to the lungs as carbamino carbon dioxide, which is carbon dioxide bound directly to an amine group of blood proteins, most notably hemoglobin. And because the rise in the bicarbonate content of the red cells is much greater than the plasma, as the blood passes through the capillaries, about 70% of the bicarbonate formed in the red cells enters the plasma. And table 35.2 summarizes the various fates of carbon dioxide. The excess bicarbonate leaves the red cells in exchange for chloride, also seen in 35.4, a process mediated by the anion exchanger 1 or AE1 or band 3, which is a major membrane protein in the red blood cell. Because of this chloride shift occurring rapidly in about 1 second, the chloride content of the red cells in venous blood is significantly greater than in arterial blood. And note that for each carbon dioxide molecule added to a red cell, there's an increase in one osmotically active particle in the cell, either a bicarbonate or a chloride. Consequently, the red cells take up water and increase in size, and because of this, plus the fact that the small amounts of fluid in the arterial blood returns via the lymphatics rather than the veins, the hematocrit of venous blood is normally about 3% more than that of arterial blood. And in the lungs, chloride moves back out of the cells and they shrink. Now, the major source of acids in the blood is through cellular metabolism. The carbon dioxide formed by metabolism in the tissues is in large part hydrated to carbonic acid, resulting in the large total hydrogen ion load. However, most of the carbon dioxide is excreted in the lungs, and the small quantities of remaining hydrogen ions are excreted by the kidneys. Acid and base shifts in the blood are largely controlled by three main buffers in the blood: proteins, hemoglobin, and the carbonic acid bicarbonate system. So plasma proteins are effective buffers because both their free carboxyl and their free amino groups dissociate. The second major buffer system is provided by the dissociation of the imidazole groups of the histidine residues in hemoglobin. So a hemoglobin molecule contains about 38 histidine residues, with each histidine containing an imidazole group that could accept or release hydrogen. Since the hemoglobin is in large quantities, it has about 6 times the buffering capacity of that of plasma proteins. Deoxyhemoglobin binds hydrogen ions more easily and is a better buffer on a weaker acid than oxyhemoglobin. The titration curves for deoxyhemoglobin and oxyhemoglobin are shown in 35.6, illustrating the differences in the hydrogen buffering capacity, showing that deoxyhemoglobin can bind more hydrogen before the pH changes. Now, thirdly, the carbonic acid bicarbonate system is the main extracellular buffer that maintains blood pH. So if the blood becomes specific with increased hydrogen, extra hydrogen combines with bicarbonate, forming carbonic acid, which breaks down into carbon dioxide and water, and the carbon dioxide is exhaled by the lungs, with the result being that the excess acid is removed and the blood pH returns towards normal. If the blood becomes alkaline, decreased hydrogen, carbonic acid dissociates and releases hydrogen, restoring the normal pH. And it's extremely effective because it's an open buffer system regulated by two organs: the lungs, which control the amount of CO2, removing excess by increasing inspiration and with a fast response in minutes, and the kidneys, which control the amount of bicarbonate, reabsorbing or generating it with a slow response, hours to days. Now, if the extra carbonic acid were not converted to carbon dioxide and water, and carbon dioxide excreted in the lungs, the carbonic acid concentration would rise. And without CO2 removal to reduce the carbonic acid, the pH would be altered from about 7.4 to 6. However, such a hydrogen concentration increase is tolerated because the extra carbonic acid that is formed is removed, and the hydrogen rise stimulates respiration and therefore produces a drop in the partial pressures of carbon dioxide, so additional carbonic acid is removed, and the net pH after such an increase in hydrogen is around 7.2 to 7.3. The Henderson-Hasselbalch equation also shows a relationship between pH bicarbonates and carbon dioxide, and the blood pH depends on the ratio of bicarbonates and carbon dioxide. Now, the arterial plasma pH is normally about 7.4, with that of the venous plasma being slightly lower. Acidosis is technically present at a pH below 7.4, and alkalosis at a pH above this value, with variations of up to 0.05 pH units occurring without untoward effect. Now, the acid-base disorders are split into 4 categories as shown here. In addition, these disorders can occur in combination, and some causes of the acid-base disturbances are shown in figure 35.3. So any short-term rise in the partial pressure of arterial carbon dioxide about more than 40 millimeters of mercury due to hyperventilation, for example, results in respiratory acidosis. As carbon dioxide retained is in equilibrium with carbonic acid, which is in turn in equilibrium with bicarbonate, the effective rise in the plasma bicarbonate means that a new equilibrium is reached at a lower pH. This can be indicated graphically on the plot of plasma bicarbonate concentration versus pH in 35.7. The pH change observed at any increase in partial pressure of carbon dioxide during respiratory acidosis depends on the buffering capacity of the blood. So the initial changes shown in 35.7 are those that occur independently without any compensatory mechanisms, so uncompensated respiratory acidosis. Any short-term lowering of partial pressures of carbon dioxide below what is needed for proper CO2 exchange, that is less than 35 millimeters of mercury, such as in hyperventilation, results in respiratory alkalosis. The decreased carbon dioxide shifts the equilibrium of the carbonic acid bicarbonate system to effectively lower H plus and increase pH. Now, blood pH changes can also arise by non-respiratory mechanism. Metabolic acidosis occurs when the strong acids are added to the blood. The carbonic acid that is formed is converted to carbon dioxide and water, and the CO2 is rapidly excreted by the lungs. Note that in contrast to respiratory acidosis, the metabolic acidosis doesn't include a change in the partial pressure of carbon dioxide. The shift towards metabolic acidosis occurs along an isobar line, which is seen in figure 35.8, which is a curved line plotted on an acid-base diagram that connects points with the same partial pressure of carbon dioxide. So in this case, 40 millimeters of mercury. When the free hydrogen level falls as a result of the addition of alkali or the removal of large amounts of acid, for example, like vomiting, metabolic acidosis resolves. In uncompensated metabolic acidosis, the pH rises along the isobar line, as seen in both 35.7 and 35.8. The two main compensatory systems are the respiratory and renal. The respiratory system compensates for metabolic acidosis and alkalosis by altering ventilation and consequently the partial pressure of carbon dioxide, which can directly change blood pH. And in response to metabolic acidosis, ventilation is increased, resulting in partial pressure of carbon dioxide decrease and subsequent increase in pH towards the normal, also seen in 35.8. Now, in response to metabolic alkalosis, the ventilation is decreased, partial pressure of carbon dioxide, increased, and a resultant decrease in pH occurs. It should be noted, however, that because respiratory compensation is a quick response, the graphical representation in 35.8 overstates the two-step adjustments in the blood pH because in actuality, as the metabolic acidosis begins, respiratory compensation is invoked and the large shifts in the pH depicted don't occur. Lastly, for completion, compensation from respiratory or metabolic acidosis and alkalosis, renal compensatory mechanisms are invoked. The renal tubule cells in the kidney have active carbonic anhydrase and thus can produce hydrogen and bicarb from CO2. And in response to acidosis, these cells secrete the hydrogen into the tubular fluid in exchange for sodium, and while the bicarbonate is actively reabsorbed into the peritubular capillary. For each hydrogen secreted, one sodium and one bicarb are added to the blood. The result of this renal compensation for respiratory acidosis is shown graphically in the shift from the acute to the chronic respiratory acidosis in 35.7. And conversely, in response to alkalosis, the kidney decreases the hydrogen secretion and depresses bicarb reabsorption. And the result of this renal compensation for respiratory alkalosis is shown graphically from acute to chronic respiratory alkalosis in figure 35.7 as well. And these are my references. Thank you.